It's a great pleasure to be here. Thank you for inviting me. I'm going to talk about the resolution and making, say, progress in this field after the Nobel Prize. This is how it was meant. Now, in the 20th century, as you know, the resolution of any light-focusing microscope, a microscope that uses just focused visible light, was limited to about 200 nanometers. Now, the turn of the century showed that at least for fluorescence microscopy, there is physics or physical chemistry that allows you to overcome the diffraction resolution barrier, and you can achieve a spatial resolution that was totally unanticipated. As a matter of fact, stat microscopy, which was the first method that proved that, allowed us to achieve a spatial resolution that is larger by a factor of 10, for example, down to about 20 nanometers. Now, as Bengt just mentioned, this has led to the 2014 Nobel Prize in Chemistry, and this is the official poster of the Nobel Foundation, showing, of course, the basic principles of stat microscopy and showing the basic principles of palm or storm microscopy, which was demonstrated a couple of years later and had a number of advantages over stat. Of course, also disadvantages, many things in common, many things different, but one thing that it clearly had in common with stat, that in principle, you could attain a spatial resolution that was at the size of a molecule. Now, if you really look at it, I'm saying deliberately, in principle. In practice, the resolution was not better than, say, resolution improvement was not better than by a factor of 10. So in practice, you couldn't get more than about 20 nanometers. Of course, there were exceptions. So with that, with a number of cases, so special fluorophores in solid state, you could get down to the single-digit nanometer range, and also with palm storm and none of special cases, but not in general. So in my view, the Nobel Foundation put it just right. Their microscopes, meaning the laureates' microscopes, crossed the threshold. And what I'm adding to this, but did not attain the ultimate limit. And so the actual title of my talk and the actual motivation for the work that I'm showing now is to do just that, attain molecular size resolution with focused visible light, of course, in 3 dimensions. Now, in order to explain to you how we get there, actually we got there, as you would see, I have to make you acquainted with the basic principles of operation of this breaking of the diffraction barrier in fluorescence microscopy. Now, especially if you're, say, high school student, you may think, okay, so why don't we get there? But you may have already learned it in school that the reason why it's not possible, say, usually to get to a higher spatial resolution than about 200 nanometers is actually very simple. It's the fact that a lens cannot focus the light down to the size of a point, of a single molecule, for example. If a lens could do that, of course, we could just direct light just on a single molecule. We could single that molecule out, so to speak, and then address each of the molecules here. And of course, we would have molecular resolution. We could collect the fluorescence, for example, in fluorescence microscopy back here at a detector. And just by scanning with this point-like focus, we could just address each molecule and have a molecular scale resolution. But that doesn't work, because light propagates as a wave. And because of that, the lens is not capable of producing a single spot of light that is a nanometer size or so, or even smaller. Rather, we'll get a diffraction-limited blob that is at least about half the wavelength in dimension. And because of this diffraction phenomenon, it's not possible to address a single molecule. Rather, what will happen is we will address all the molecules that fall within this spot size range, and then all of these molecules will kind of be flooded with light. They will light up and produce, of course, backscattered light. Now, in our case, the backscattered light is fluorescence light, so light of a slightly longer wavelength, which is collected here in the detector. And now you can imagine that each of the molecules, of course, also produces a wave of fluorescent light that is also diffracted. And even here at the detector, of course, we will get this diffraction blob for sure. And then each of the molecules here will produce a blob of light that will inevitably overlap in space due to the finite size of the spot, of course, of fluorescent light in the detection range. And as a result, it's not possible to tell the molecules here apart at the detector. Now, the solution to this long-standing problem in the end was very simple. Guess what it was? Just make sure that not all the molecules that are covered here by the light are in the end capable of scattering light back. In the case of fluorescence, you have to put them in a state, in a molecular state, in which they are not capable of producing the light back. So in simple words, you can say you have to find a way to turn some of the molecules off. For example, turn all these molecules off but this one, and then you can separate this molecule from the rest. And then, of course, you can behave, the system can behave as though it would have addressed just this molecule. And then you can sort out this mess in here, and you get a signal just from that molecule. So actually, if you will, the basic element that really created the field and still is the foundation of the field is to implement an on-off transition here in the focal region so that not all the molecules that inevitably flooded with the green light are capable of producing signal at the end. Now, in STAT microscopy, without going into details, this phenomenon that turns the molecules off is called simulated emission. It just kicks the molecule down to a ground state in the case it gets up, so it keeps the molecule constantly in the ground state, keeps it off, so to speak. But this is just one way of doing it. There's many other ways in many other molecular transitions that allow you to turn molecules off. So this was just the starting point. And there are also other mechanisms, for example, like cis-trans isomerization or electron transfer and so on, to do the same thing. But in the end, it's on-off. Now, STAT microscopy has the hallmark that we use a beam of light that turns molecules off. And this is why I'm showing you this red little thing here. It is a beam that has a hole. And wherever there is light, the molecules are turned off. And if there is no light, and this is the case at the very bottom of the hole, it's a donut-shaped beam, the molecule is not turned off, and then, of course, you single out that molecule, for example, and so you can separate this molecule from the rest due to this on-off transition. Okay, now Palm Storm. What's the difference here in Palm Storm compared to STAT? Again, you play on-off, so that's the same thing, but the way the on-off is played is substantially different. Here, a molecular transition is chosen, such as a cis-trans isomerization or so, that turns just one molecule on within this subdiffraction range, and the rest is off, and this is how we can distinguish this molecule from the rest. But purely stochastically, you could do it by beam of light, where every now and then there comes a photon that turns molecules on. You can do it thermally. There is a way of association and dissociation. There's many ways of turning molecules on and off, as I said. But now, of course, if it's purely stochastical, there's no way to know which of the molecules have been turned on. So it could be any of these molecules that I've shown here. And of course, you don't know where this molecule is located. And therefore, you have to find it out. And this is possible if you place a detector in here that has many, say, detection points, an array of detectors that gives you a coordinate. And that's the first condition. And the second condition is that many photons are emitted from the molecules consecutively so that you can associate these photons to that particular molecule in the on state. And now, if you're getting, say, n photons, 10, 100, or 1,000 photons from that molecule, you will get a pattern of light in here. And then you know, of course, that the center of that pattern must be associated with the position of the molecule, because there, where the pattern is brightest, of course, this must be geometrically just related to the position of the molecule. And so what you do is you find out the position of this on-state molecule that you are detecting just by calculating the centroid of this diffraction pattern with a photon number n. So n is like 10 or 100 or 1,000 or even more photons. And the more photons you have, of course, the better you will know the position of the molecule. Okay, so this is the concept of Palm, and if you compare it now with the concept of STAT to see the similarities and the differences, as I said, on and off, this is the basis of both. But the difference, of course, is in the way you find out the coordinate. Here, of course, you need many photons to find the position of the molecules, and here, of course, you need many photons to define the position of the molecules. Because the brighter this donut beam is, the better defined will be the zero in here, and the better defined will be, of course, the position at which you know that the molecule will be located. And it's very obvious. If you have just one photon within the diffraction zone, just one photon, imagine a beam of a single photon. It could be anywhere, go anywhere within the diffraction zone. There's no way to create a pattern or define a position. But if you have many, the more you will have, of course, the better you can define the zero in here or the position in here. So the secret to having or to getting a position optically with photons is to have many photons. But the way it's done, as I said, is different. Here the many photons come from the laser. They define the position here. And here the many photons come from the dye. And this is a big difference. Why? If you have to do the coordinate with the many photons that come from the dye, you have to rely on the fact that the dye is bright and emits many photons. So this backscattering of fluorescence is very, very strong. But fluorescence, I can tell you, is not something reliable. It is very unreliable. The dye bleaches, the molecule goes to a dark state. So doing it in this way, say with many fluorescence emission, is not very reliable. And this was the reason why Palm Storm never got beyond the 20 nanometers, because at some point the fluorophore didn't emit enough light, and it takes, of course, a lot of time to collect all these photons, and by the time the molecule has moved, and this is a problem. But here, you have zillions of photons. They come from the laser. No problem in putting in as many photons as you want. The laser basically gives you an infinite number, almost infinite number of photons, because there's no shortage of photons from a laser. And you can perfectly define the position here of the zero. So if this is the case, why not combining the strengths of the two methods, so to speak, and come up with a method that is a synergy of the two? And what is the strength now? Well, it's very obvious. Palm already deals on the separation level when it comes to separating features on the single molecule level. And there's no better separation than separating single molecules. There's an advantage. As I said, the disadvantage is the way the position is found out. Here, of course, the strength is to find out the position. So why not combining the two, say, doing the separation on a single molecule level, like in the Palm concept or Storm concept, but doing the positioning in the way it's done instead, with the zillions of photons that come from the laser. And if you combine the two, you must get a higher spatial resolution and better pictures. So that has been the logic behind this. Now, in the case you didn't really listen to my talk for some reason, I don't know, maybe you were interested in the way I look or whatever, or my baldness or so on, you can start now from scratch, okay? I'm explaining everything from scratch, and I'll make it in an easy way. Let's assume now we have turned on the molecule, and this is the molecule. Play on off, we have turned on just one molecule. Now, what people usually did, also for the Palm Storm Nobel Prize winning concept, they had so flat illumination of light in the focal plane of the microscope. You see it here. This is the objective lens, a flat illumination of light. And then this molecule produced fluorescence. It was focused on a camera. This is the camera, the pixelated detector, so to speak. And then, of course, the fluorescent light that is shown here on the monitor of the computer is diffracted because of diffraction. So here, the lens, of course, produces a diffraction blob of light here on the camera. And then, as I said, people understand by localization, calculating the centroid of that pattern. And as I said, the more photons you have, fluorescent photons you have, the better you will know the position of the molecule just by calculating the centroid. It scales with the inverse of the square root of the number of photons. So if you want to have a 10-fold higher spatial resolution, you need 100-fold more photons. Okay, but this is not how we're going to do it, because we're doing it now, the localization, in the STAD-like way. So we're going to use a pattern of light. We are going to use, so to speak, a donut. The donut is very good because the donut itself, or the donut zero to be precise, perfectly defines a coordinate in the focal plane. So you see, this is the maximum of the donut. Here there is a zero. So here in the intensity 0, there is no light, and this is perfectly defined because we have so many photons from the laser that perfectly define, say with sub-angstrom precision, if you will, the position of that zero. Not only that, we can measure if the fluorophore is right at the center of the donut, because if the molecule is right at the center of the donut, it cannot emit light because there is no light. It's a zero intensity. So if the molecule is slightly off, it will emit light, and then we will know, oh, it's not at a zero because there's some light coming. And this helps us, of course, easily to find out where the molecule is actually located. Now, since my last name is Hell, and we spoke about demons this morning, I said, okay, I have to introduce a little demon. So this is my little demon. And this little demon has a piece of information that humans don't have. And in this particular case, this little demon knows where the molecule is. And let's assume now for the moment, we do a Gedanken experiment, a thought experiment, as it is done with demons, that this little molecule moves, and the demon, knowing where the molecule is, shifts actually the excitation beam in such a way that the zero of the donut always coincides with the molecule. Okay? So what does that mean? If the zero of the excitation beam donut always coincides with the position of the molecule, of course there will be no fluorescence emission. That's very clear. Not only that, the demon can perfectly trace, of course, the position of the molecule simply because the actual, say, actuation on the beam deflector, or the position of the donut, would be identical with the position of the molecule. So the demon can perfectly trace actually the position of the molecule. So what can we infer from this? In this Göttingen experiment, of course, the demon can perfectly localize the fluorescent molecule without requiring any single fluorescence emission. Without any detection of photons or anything, there's no fluorescence emissions going on because the zero of the donut perfectly coincides with the position of the molecule. And this is contrary to the normal localization where you need many photons. As I said, n photons, 10, 1,000, 100 times is even better to get a factor of 10. As I said, the more photons you have, the better localization, but here you don't need any fluorescence emission in this thought experiment. And then there comes a shocking other conclusion that might surprise you, especially if you're trained as a physicist. Will there be a wavelength dependence here? Not really, because if the molecule is slightly off the zero, who cares what the wavelengths of this donut is? So there is no wavelength dependence, say, in this limiting conceptual range. And so this means that the idea, so to speak, of localizing the molecule with a pattern of light, in this particular case with the donut, is very profound. I'm going to show you this now. Now, in reality, there's no demon, of course, and also, even if I'm named Hell, I have to work with something that works. So we have a controller, we have a detector, and of course, we have this beam deflector that can shift the donut around. And then what we do is actually we measure the fluorescence that is produced if the donut is slightly away from the position of the molecule. So if the molecule starts moving, of course, we of course try with the controller to shift the donut excitation beam just on the position of the molecule so that the zero, of course, coincides with the position of the molecule, but we will fail because we're not demons. But what we will see, we will get a detection of light, so fluorescent light, that is measured here. And that, of course, is fed into the controller. And this fluorescent light gives us actually a clue where the molecule actually is. And we can use, of course, this information to reposition, of course, the donut. But we have to do this only by a little, of course, because, say, if it's slightly away from it, you don't need many photons, so to speak, to find out the position of the molecule. And this, of course, helps you to find out the position of a molecule here with a minimum amount of fluorescent photons. Why? Because the fluorescent photons that are detected in here are just used for refinement. You don't need to have 100, 1,000 or so photons in order to get a position with a certain precision. Okay, now you will ask, okay, how do we do that in practice? How do we know where the molecule is located, or what does the controller actually do, in other words? It's very simple. Now let's assume the molecule is in here, okay? And we don't know where the molecule actually is, so we put it in here in this view graph. We assume the molecule is somewhere within this range, within this line. I'm just assuming one direction because it's easy to explain the concept in a single direction only. And now, it's very obvious what we would have to do. We would have to scan this donut across the molecule to find out the position where there is no fluorescence emission, because then we know where the molecule is, because there, the zero of the donut, whose position we know, of course, must coincide with the position of the molecule. So this is the intensity profile of the donut, and this is the fluorescence that we get. Now, let's move, and you see, now, the zero is there, and the fluorescence is minimum. And in this way, just by scanning, of course, with the donut across the molecule, we find the position of the molecule, we know where it is. Now you say, oh, this is a quite tedious thing, because you have to scan all the time to find that little position. No, you don't have to do that. Because if you know that this is, for example, a quadratic function, and usually the zero in first approximation is always a quadratic function, it's perfectly enough to know the endpoint, say, of the signal from the endpoint, so the counts of fluorophores that you have at the endpoint of the two traces of the trace, and 0 and 1. And then, of course, you can extrapolate, because you know this is a quadratic function, on the actual position xm. It's very simple. It's basically algebra, 7th graders algebra. You can easily calculate that. So if you assume that you have a quadratic approximation here of the intensity at a donut. You can just, say, invert a simple quadratic equation, and you will find out that the position of the molecule is given by this distance L, in which you assume that the molecule is located. Usually that's a diffraction range, say 200 nanometers or so. And you just take the ratio of the two endpoints, of course, of the fluorescence of two endpoints, and then instantly the position pops out. So if the two are equal, of course, xm is in the middle. Now, this simple equation is actually surprisingly profound, because one thing it's telling you that there's no wavelength dependence. Gone. There's no wavelengths in here. So, no. You just find it out by ratioing the brightness of the two endpoints, of the measurement at the two endpoints. And not only that, there's no dependence on the molecular rotation. You will know that usually, I mean, this fluorescence emission is a dipole emission, and then the orientation of the molecule matters, of course. And this is a big problem for this palm store concept, because due to this, the resolution can be degraded by 10, 20 nanometers, even more, because the centroid of that diffraction pattern is off by 20 or 30 nanometers. No one has sorted out this problem yet, so it's all kind of sometimes pulled under the carpet, but here it's inherently gone. Because the only thing we do is we inject position information with the donut. The donut injects position information, and then finally we take this ratio, and that's it. Now, of course, there is an uncertainty still left with xm. It's not perfectly defined, and that will depend, of course, on L and the range of uncertainty that we have to begin with, and of course, on the brightness of the molecule. So the higher the number, and 0 and 1 is, of course, the more precise the sigma will be, so the smaller sigma will be, and the higher the precision, hence also the spatial resolution that we get. But the good thing now is, and that's the fundamental difference to the concept before, here it scales linearly with L. The square root of n is still there because of Poissonian statistics. But here, you have a handle on it because the uncertainty scales with this range L. That means once you have coarsely estimated, oh, it's not within the 200 nanometer range, it must be somewhere here with 50 nanometer uncertainty, you just scale down L. And this is, of course, more effective than waiting for more photons, as you do in Palm Storm, because this scales with the square root, which is not so good. It is damned. As I said, for 10-fold higher resolution, you need 100 times more photons. But here, if you decrease L by 10-fold, you get 10-fold increase in spatial resolution. So it's more effective to zoom in on the molecule and then do the same thing again. And so you can iteratively actually, say, so to speak, approach the position of the molecule, thereby reduce the number of fluorescence photons that you need to detect in order to find out the molecule with a certain precision. And this is why this concept is so effective. Okay, now, of course, this was in one dimension, how you do it in 2 dimensions or in 3 dimensions. If you do it in 2 dimensions, of course, you need at least 3 points. It's better to use 4 points to avoid ambiguity, so that's what we did in practice. So measure here, with the donut zero placed here, 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 and here. Take the 4 numbers, and then the equation is not that simple anymore. It's a more complicated function, but in the end, it boils down to the same thing. You see here the uncertainty, which boils down to, or comes down to the resolution in the end. Again, you scale linearly with the range in which you anticipate the number of molecules, and of course, square root of n, that's there, but we can outperform the square root of n behavior just by zooming in, so to speak, on the molecule, by making the L smaller, and then and quickly finding out where the molecule is by overlapping, so to speak, the zero of the molecule, at the zero of the donor with the molecule. Okay, how does this compare with a normal, say, camera-based localization? And you see it here. So this is the number of photons that you need, and this is the precision that you get. This is for a realistic camera. This is actually a measurement, and now you see with 10 photons, you get, in our case, we call it MINFLUX because it has this minimum in here and produces a minimum of fluorescence flux for attaining a certain precision. You need only 10 photons. With only 10 photons, you get 10 nanometer spatial resolution. But if you then try to do the same thing with a camera, you would need 29 times more photons, so 290 photons in this particular case. So we are more efficient in terms of the fluorescence photons that we need. And this is actually a theoretical calculation, so-called Kramer-Rauerbaum. Kramer was a Swedish mathematician. You see here, this is the equation for the uncertainty that comes out. Now, again, zooming in on the molecule is the key, because this helps you getting up the precision without requiring more photons. But in the end, of course, you have to prove that you get better images. I'm going to show you this. Again, this is just a conceptual description. In order to separate the molecules, let's assume this was an object, we have to turn the molecules on and off, so this has not changed since that. Again, but it's done on a single molecule, say, level, as it is done in Palm or in Storm, and then we localize the molecules and then get a picture. Or you can also do tracking, and you will save the number of fluorescence photos that you require in order to track the molecule with high precision. Now, this is now a cartoon showing, say, fluorescent molecules at very small distances. You see here 11 nanometers or so. At the time the Nobel Prize was given, this is a Palm Storm recording, these molecules could not be separated. Neither with Palm Storm nor with that, because they are too close. The resolution was not there. I said about 20 nanometers was the maximum resolution. Now with this MINFLUX concept, you take them apart. You turn the molecules on individually, locate them with the donut, and here you have it. So basically, we are down to the range that is the range of the size of the molecule, which is of the order 1-2 nanometers. There's a link or so, but it's basically down here. 6 nanometers, so distances. Okay, again, Palm Storm cannot resolve it. MINFLUX takes it apart. So in other words, take into account the fact that we use just focused visible light. We have reached molecular size resolution, essentially, with focused visible light. I mean, 25 years back, I think it's fair to say, if someone just stood here and say, oh, one day, not very long from now, so to speak, 10 years back, 15 years back, one will be able to attain molecular scale spatial resolution with focused visible light. People said, this guy is totally nuts. This is not going to work. No way, but here it is. Not only that, of course, you can take large field of view data, of course, with high spatial resolution, because you can scan the donut over a large field of view. Why not? And then you zoom in on each molecule that is there, and I'm going to show you now large-scale pictures. And here, these are, again, nuclear pores, similar to what you've seen on the Nobel poster. But now, you see, basically, this is the resolution that we have. Essentially, these are individual molecules here, labeled molecules. Again, very nice, very nicely resolved. And as I said, the spatial resolution is on the range of about 2 nanometers in sigma or so. I compared it with that, which won the Nobel Prize. And I'm comparing it with confocal microscopy, which I shouldn't do. Honestly, I feel sorry about those people who still use confocal microscopy, but, well, that's it for the moment. And you see now the evolution that has happened. It's here, between here and here, it's a factor of 100. So it's quite a difference. And now, again, I'm showing you this was also with fluorescent proteins. Here you have the data, so the median here of localization precision is 2.3 nanometers in sigma. Not only it works for fixed cells, it also works for living cells. That's important. This was done by focusing into a living cell on the nuclear envelope, again with a fluorescent protein. And also works in 3D. So biologists want to have something that also is obtained inside the cell in 3D. Now, this is 3D data. The color encodes height, by the way, but you see, say, the impression of height in Z very, very clearly. Again, the Z resolution is as good as the X and Y. You see here, 2.6, 3.1. Is another rendition in 3D. So this is a nuclear envelope, and it's showing again that this is true 3D data. Now, you say, if it's spotty, it's because it's molecules. And the challenge now is for the real chemists, of course, to do fantastic labeling that really labels every molecule that you want to see. So the challenge is now basically with the organic chemists to get good labels or to do the labeling perfectly right. But from the physics, from the concept or so, I think this matter is virtually closed, if you ask me. Having reached now this resolution, it's very obvious. But of course, biologists say, can you do 2 colors? Of course, we can do 2 colors. This is showing you two different types of fluorescent molecules, and this is a 2-color recording. Of course, we can also do 2 colors in 3D. This is engineering, if you will, if you see that from a scientist's perspective. But it's important engineering, because in the end, the biologists want to use it and they want to get pictures in 3D. They want to find out something biologically interesting or medically relevant, but I'm sure they will, because the resolution increase is now so much over the diffraction barrier that I have no doubt that major discoveries will be made in the life sciences as a result of this fundamental change that has happened in the last 20 years. And so this is a 3D rendition, of course, of a certain protein in the synapse. And definitely one of the applications that will come in the next future is that you really sort out the molecular maps of anything you can imagine, the nucleus, the synapse, the immunological synapse, whatever. So there's many other things. There is another important aspect. Because you need fewer fluorescent photons, you can also image much faster. This is also important. And I'm showing you this now. We made a little experiment where we attached a fluorescent molecule rigidly to a surface with a piece of DNA. And this is a localization now down to 2.4 nanometers by measuring 4 points in here. And then, of course, we took another type of molecule that was allowed to move. Okay? Not quite like that. It's just a cartoon. I couldn't make a random movement. In fact, it was a random movement. And then, of course, the localization spreads out over here. You need much fewer photons to get the same, say localization precision. So you get a time resolution for the same precision of localization that is substantially better, order of magnitude better. And this is just the traces for the experts of you to show you that this is clearly vindicated, this movement. And this is now not just a random cartoon. This is the actual movement of that molecule in this focal plane. So it really moves like this. And so the reason why I'm showing this, so one can imagine now experiments at a very high speed. So you label something like a protein, and the protein starts to fold or something. You can get the movement within a short period of time with very high precision, with nanometer precision. No way to do that with a camera, because the camera you have to wait until 1,000, 2,000, 10,000 photons come in order to find out the position. But here, using this donut that defines already a position and use the fluorescent photons only for refinement is so fast. This will open a new area of application. So summarizing this, the resolution is basically down at the molecular size. And if you calculate molecular movements of 10 microseconds, so within a short period, should be possible in a 10 nanometer range with 1 nanometer precision. And if I was a biophysicist and maybe 30 or 25, I would just go for this, because there is plenty of things to discover, because this dynamic range no other method can offer, and in a living cell condition. This is important. Now, as I said, there are new fields of application like molecular maps or looking into molecular dynamics and so on. But the good news, and here, of course, I'm biased. I say, speaking very openly, I have shares of this company. This company has released last week, actually, a commercial version of these systems. Of course, they do get the same resolution as we do get in the lab, because it was founded, actually, by my former PhD students, postdocs. And, of course, they have a high spatial resolution. This is a lipid, actually, in a lipid bilayer. See how quickly you can actually see this movement of the lipid. And in essence, you can buy it and you can use it. I think this will speed up, of course, the application of this new method. Now, for the physicists, a few insights. There is no optical nonlinearity used in here. So when I was a student in the 80s, I knew, of course, that using some nonlinear optics, you can overcome the diffraction barrier. But this was kind of leading the wrong direction. There's no optical nonlinearity used in here. And the second thing, there's no dependence on the wavelengths of the resolution. If you think the way it works, there's no dependence anymore. Because what you do, actually, you match actually two singularities. One is the molecule, and the other one is the zero. And in the first approximation, the wavelengths, as I indicated with the thought experiments, with this little demon, doesn't really matter. And there's also something very heretic, if you think about it. No wavelengths dependence in a focusing light microscope and resolution. I mean, this is crazy. Anyway, it shows science is crazy at times. So with that, I'm acknowledging the people who have really contributed to this development. I don't have a big group anymore. Fortunately, these two guys left already. They went to industry. So these are still in my lab. Actually, Yasmin Pape is a PhD student, and he's Francisco Balzarotti, a clever postdoc who is now becoming a professor in Vienna. They've done great work, of course. And of course, also me, I'm looking for clever people who are motivated to make a fundamental difference in science. Thank you very much.